Hey, what's up, you guys? Yes, the cell cycle.
Cancer is a genetic disease that's caused by changes to genes that control cell function. The mutated genes cause unchecked, accelerated mitosis. The cells divide uncontrollably, spreading into surrounding tissue and stealing nutrients.
The cell cycle begins with interphase. This stage takes up about 90% of the cycle. Interphase is made up of 3 stages: G1 phase, S phase, and G2 phase. During G1 phase, the cell grows physically larger, copies organelles, and makes the molecular building blocks it will need in later steps. In S phase, the cell replicates the DNA in the nucleus. It also duplicates the centrosome, which helps separate DNA during M phase. During the second gap phase, or G2, the cell grows more, makes proteins and organelles, and begins to reorganize its contents in preparation for the mitotic phase, or M phase, which is usually the shortest part of the cycle.
Mitosis produces 2 genetically identical daughter cells.
In prophase, the chromosomes start to condense and the mitotic spindles begins to form. Its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. The nucleolus, a part of the nucleus where ribosomes are made, disappears. This is a sign that the nucleus is getting ready to break down. In prometaphase, the chromosomes finish condensing and the mitotic spindles organize the chromosomes when the nuclear envelope breaks down. The mitotic spindles grow more and some of the microtubules start to capture them. In metaphase, the spindles have captured all the chromosomes and lined them up at the metaphase plate. At this stage, the 2 kinetochords of each chromosome should be attached to microtubules from opposite spindle poles. In anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell. The protein glue that holds the sister chromatids together is broken down, allowing them to separate. The chromosomes of each pair are pulled toward opposite ends of the cell. Microtubules not attached to the chromosomes elongate and push apart, separating the poles and making the cell longer. Here's a close-up of the sister chromatids splitting apart. In telophase, the cell is nearly done dividing and starts to re-establish its normal structures as cytokinesis takes place. The mitotic spindle is broken down into its building blocks. 2 new nuclei form, 1 for each set of chromosomes. Nuclear membranes and nucleoli reappear. The chromosomes begin to decondense and return to their stringy form.
Cytokinesis, the division of the cytoplasm to form 2 new cells, overlaps with the final stages of mitosis. It may start in anaphase or telophase depending on the cell and finishes after telophase. Cytokinesis pinches the cell in 2 like a coin purse with the drawstring. The drawstring is a band of filaments made of a protein called actin, and the pinch crease is known as the cleavage furrow.
Meiosis produces 4 genetically diverse daughter cells. This occurs in the gonads and produces haploid sex cells, and then in fertilization it becomes diploid again.
In prophase 1, chromosomes condense, paired homologs connect to each other during the synapsis state. Crossing over begins during pairing and synaptonemal complex formation and is completed while the homologs are in synapsis. Similar to mitosis, nuclear membrane fragments, chromosomes become visible, and centrioles move to opposite poles. Crossing over is the process in which homologous chromosomes pair up with each other and exchange genetic material. In metaphase 1, pairs of homologous chromosomes line up at metaphase plate. The alignment is random. In anaphase 1, homologs separate into chromatids. They go to their respective poles, guided by a spindle apparatus. In telophase 1, nuclear membrane reforms around each set of chromosomes. In cytokinesis, cell splits, resulting in 2 daughter cells. This marks the end of meiosis 1, which as you can see yields 2 non-identical daughter cells. To form haploid gametes with 23 chromosomes, each in humans, the same process starts again and goes through meiosis 2, this time resulting in 4 haploid cells. The normal number of 46 chromosomes will be restored during fertilization when a sperm and egg combines. 4 haploid daughter cells are all genetically non-identical.
Thank you for giving us your undivided attention.
Crystal, you're so happy, funny.
And I hope this was helpful.